From Microbe TV, this is Immune, episode number 75, recorded on December 19th, 2023. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, everyone. Welcome back. Ithaca is now, winter is on its way. It's been a little warm, but now we had a little snow, so it's nice. I know, Brianne, I think you're coming up this way, so you're checking on the weather. Should be not too bad. I don't know if we're going to have a white Christmas or not. It's up in the air.
We just had a wet Christmas. We had like 3 days of rain here.
Yeah, we had flooding
It's a
too.
mess. Also,
there's a lot of water.
Joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. Yes, we had all sorts of rain here as well, but it seems kind of nice now, so that's good. And hopefully I won't have to drive during snow.
And today we have a guest for you from the Harvard School of Dental Medicine, Ung Shui. Welcome to Immune.
Thank you so much for the invite. I'm very excited to be here.
Yeah, thanks for joining us. We're excited to talk about your work. Very cool stuff. And lately we've been having people on who did the work, right? Cindy and Brianne, I think that's cool.
Yeah, absolutely.
Yeah. Who can talk about it better than them, right? That's the whole idea. Before we get started, don't forget we depend on your support to do these programs. And that means financial support, not just, mental support or whatever. That's great too. But
Emotional support is always useful.
It's all good, but we need your cash. Cold, hard cash. Sorry, folks. That's the reality of the world. However, we are a 501c3 nonprofit, so your donations are U.S. federal tax deductible. You can go to microbe.tv/contribute. And Cindy, you wanted to make a few comments about an initiative as well.
Yeah, so I am part of a group called Solving for Science, and it's a new community of scientists and allies working to change the culture of science from within, so tackling really big problems. And in 2024, they're going to tackle something really big that I think everyone can agree is broken to some extent, and that's publishing. So we're looking for a lot of immunologists. So my fellow immunologists in Solving For developed an exciting new pilot to test. So it's a new journal-free system, and preprints will undergo what we're calling a peer improvement review, which reframes the review process as an opportunity to make papers better, while also quantifying a paper's potential impact and quality through a transparent scoring system. So at the end, Solving For is hoping to compare how this process stacks up to regular journal reviews and publishing. So what we need are immunologists with preprints and PIs as reviewers, so they can be part of this groundbreaking shift towards a transparent, community-driven platform for storing and accessing all of our discoveries. So you can learn more about this at solvingfor.org. So that's solvingfor.org/DSDC-pilot, PILOT. And there you can sign up and you can learn more about it. You can be a reviewer, or you can have your paper reviewed as part of this pilot program.
All right, we'll have a link for that in the show notes in case you missed it.
Cool. I'm definitely going to look into that.
Yeah.
Yeah.
Participate.
Sounds like a great initiative.
They're going to do this parallel process where you're still going to submit your paper and have it reviewed and published normally, but they're also going to have this other mechanism in parallel and see how they compare as far as impact in the long run.
Well, I wish them luck.
Yeah.
I can't imagine what they're going to do next. They're going to tackle peer review.
So other things that we're tackling are diversity in science and science communication, which is near and dear to your heart.
Yes. Okay, Ung, tell us a little bit about yourself. We want to know all about your history, as far back as you can remember.
Sounds great. So I'm Chinese Canadian. So I was born in China. That's as far as I can remember. I was born in China, and I moved with my parents to Canada when I was 13. So I stayed in Canada until after I finished college. I went to college at the University of Toronto. I studied electrical and computer engineering. And I was doing research at the same time. That's my first exposure to research. I was in a machine learning and computational biology lab. So at the time, my coursework was mostly focused on electrical engineering, and we did a lot of AI, ML in the class. So the research was very great environment to apply the knowledge to a real world setting. And at this time, my immunologist came to collaborate with us, and she studied juvenile idiopathic arthritis, which is a very terrible autoimmune disease that happens in children. And so I was working with two different labs, one doing machine learning, the other one doing immunology. And I had to kind of bridge these two fields as an undergraduate student. So that I just felt like, there's so much to learn in both disciplines. So that was the first time I decided that immunology was really cool. It's involved in every disease, every major disease out there. And there is so much complexity in the immune system. We don't understand it enough. But it seems like big data approach, like the engineering approach that we're using, was promising to gain a better understanding of the immune system. So I started from there, and I came to the US for graduate school. I initially came to Yale. I did a master's in bioinformatics and immunology. And then I came to Boston to do my PhD in the Harvard-MIT Health Science and Technology Joint Program. And I did a short postdoc and started my lab this year. I joined the faculty here January 1st.
Awesome. So you're coming up on your 1-year anniversary.
Yeah, that's right. We just had a holiday party for lab turning 1 year old.
Oh, wow.
That's awesome. What types of things did you work on as a grad student? I'm trying to imagine your transition from sort of more of a machine learning type of project into immunology. It sounds like you started that as an undergrad, but then what types of things did you do as a grad student?
Yeah, that's a great question. So initially at Yale, I was still doing a lot of work on somatic hypermutation. So somatic hypermutation, as we know, is like a B cells gaining mutation at a very high rate, which is how B cells develop diversity, which is how antibodies are able to diversify. And it's a very interesting computational problem. We generate a ton of sequencing data. And we were just building clonal trees to trace the lineage of each clone. And that was a very interesting problem. And one of the findings that I learned from that project is that males and females seem to have different somatic hypermutation patterns.
Huh, really?
Yeah, so we all immunologists know for a long time from clinical observations that males and females have different levels of immune responses. Males are more likely to develop severe symptoms to infections, as we all learned from COVID. And on the other hand, females are more likely to develop autoimmune diseases. But the molecular mechanism that explained this diversity was not understood. And our analysis of big data provided one explanation. So we analyzed lots of mutations from lots of sequencing data from B cells. And we found that one pathway, the somatic hypermutation repair pathway, it seems to be biased between male and female, especially as they age. So this, we think, is an explanation for when males get older, their antibody diversity and affinity to pathogens probably also, there's a difference. So that was one of the pretty exciting findings from my work back at Yale when I was doing somatic hypermutation. We also built somatic hypermutation repair models so people can use our database to look up the mutation patterns. And my undergraduate work also led to some interesting findings. So we were using machine learning to study juvenile arthritis, and it was also to develop a novel machine learning algorithm to understand how different patients respond to therapy. And we found that even though juvenile arthritis is a group of heterogeneous diseases, we were able to find a molecular signature that is common to a lot of patients, and that is predictive of therapeutic response. So we also published that paper. And so we were like at that time, I already knew that I definitely wanted to do immunology, and we know there's a lot of like cross interdisciplinary research, but it was very obvious that if I really wanted to make true contribution to immunology, I have to know the field inside out. So at Yale, that gave a really great opportunity. So as you know, Yale has excellent program in immunology, and I was taking classes with very prominent immunologists like Dr. Akiko Iwasaki, Ruslan Mechitov, David Scharz, Susan Kaig, Carla Rothen. Like I didn't even know how lucky I was to study with these folks. But yeah, when I look back, very, very lucky, and I really learned immunology. And my mentor, Dr. Steve Kleinstein, he is like the best computational immunologist at Yale. And I think I got really good training there. So at this point, this is still not yet my PhD work yet.
Oh no, that's okay. This is really interesting.
This is great.
Yeah. So at this time, I already published 3 first author papers. And I was like, if I wanted to just finish a PhD in a couple of years, that shouldn't be terribly challenging. But then I was thinking like my original training is electrical and computer engineering. If all I want is to, just find a job or, like I could easily just go to the software industry and work. The reason why I want to do research is to really be able to use the unique skill set to, make a major contribution. So I decided, when I did my PhD, I was in Dr. Nir Hacohen's lab at the Broad Institute of MIT and Harvard. So he is a leader in systems immunology. And I just really wanted to work on some very ambitious projects. And I got an opportunity to do that. So that was very exciting. So the cytokine dictionary that is one of my projects from my PhD. So this
is the publication in Nature, right?
That's right.
Just this year, just a couple of weeks ago.
That's right.
And the title is Dictionary of Immune Response to Cytokines at Single Cell Resolution. And that's what got you on my radar, because I saw this and I was like, what? And I started looking at the paper and I said, this is, wow, a lot and so incredibly useful for the field. So how did you come up? Go ahead,
I have
Brianne.
one other sort of training or getting to this point question. So I teach and work with a lot of students who are sort of thinking about next steps. And I'm curious, when you think about your training, how much time, if any, do you spend doing bench science versus doing more sort of computational work? And how did that change over time, or how do you feel about that? What's going on in your lab on that front now?
Yeah. So in my lab, it's 50-50. We have half of the students working on computational side and half of the students doing experiment. And in my own training, in my own time, I also try to do a balance of both. So initially, when I first said, I wanted to do experiments. So I started to do experiments in undergrad, but people were like, but computational, it's not common to do both. You shouldn't be doing both. You should really just do one. And I said, no, I definitely want to do both computation and the experiments. And I think that was a really good decision. I mean, I had a lot of pushbacks back then, but then I persisted and I learned both inside out. And that was very helpful. So I definitely recommend for anybody who wants to do interdisciplinary research, it's important to not just learn a bit about a subject. It's like just so important to go deep into it because, until you get very deep training in both sides, it's very hard to understand the nuances. Like for a lot of experimental data that I was working with before, it's just really hard to understand like the specific experimental procedure that could lead to certain type of outcomes, and you only know it once you do it. So I highly recommend it. Like if anybody wants to do both computation experiments, go for it.
Awesome. That's really helpful.
Talk about
and related to that. Oh, go ahead, Vincent.
First, I just want to say that was an ambitious project.
Yeah. Well, that was what I was going to follow up on. I was shocked there's not that many authors on there for the amount of work that went into this project. So can you tell us a little bit about, how did this come about? So if I remember correctly, it's like 68 individual cytokines.
86.
So tell us, 86. Sorry, 86. Got it backwards. So can you tell us about that? So how did you do this? How did you come up with this? And how did you actually make this happen? Because this is an incredible number of mice, an incredible amount of single cell analysis.
Yeah. So I was already doing a lot of work with mice at that time. I was optimizing procedures to study vaccine responses in lymph nodes and the cancer immunotherapy. And it was kind of an anecdotal finding that the lymph node is much more diverse than what was known before. Like people used to think lymph nodes are just, they're B cell follicles, T cell zones, maybe there are capsular cells, macrophages, and dendritic cells moving in. That's the textbook diagram for what lymph node looks like. But when I depleted a lot of B cells and T cells and then ran single cell RNA sequencing, I learned that there are so many other cell types in lymph nodes, like, NK cells, PDCs. You can find them in literature, but it was not really not everybody was thinking about it a lot. So once I saw this procedure, and then we decided to do an ambitious project, we just decided to systematically measure every cytokine we could buy out there. So the project was actually inspired by a completely different field. So we, my advisor and I and a couple other people from the lab went to a conference, and we attended a talk by Dr. David Baker. So he is a very big person in protein structure prediction. That was like 5 years ago. Now AlphaFold can predict almost any structure, but back then that was groundbreaking. We attended his talk and we learned how his algorithm could predict structures of proteins. And we were like, oh, this is mind blowing. How did he do that? And I even tried to talk to him afterwards. I was saying that, what exactly did you accomplish to, be able to do this? And he was basically just saying that, after he started his lab, he just started small and then, predict some simple structures first. And then there are more and more graduate students coming in. Everybody contribute a little bit more and eventually just build up this full capacity. And that's why he was able to show us those impressive results. So there was, so that's the time that I realized, I also want to do something important for the next 5 years. It doesn't matter that I start small or something, but I want to aim towards something truly important. So my advisor and I talked that we can basically build a big database of mice responding to different cytokines. And that was, it is very large scale, even by today's standard, because it's 17 cell types responding to 86 cytokines. So it's 1,400 cytokine cell type combinations. Even for single cell, it's so popular today. Now, a lot of labs are doing it, but this scale is still very, very large. So we're definitely going very ambitious in this. But I knew I was like we knew we were able to do it because, I initially got my training in computer engineering. So handling this large amount of data wouldn't be a problem. So we basically just decided to go for it, and we did it.
I noticed you submitted the paper 2021. Oh my gosh. They give you a hard time?
No, it was very smooth.
A 2-year very smooth process.
That's another story, but that was I noticed that in Nature they put a date on the front page. It's extremely obvious.
Yeah.
So everybody asked me this question. They're like, the reviewers asked really hard questions, but no, actually it was very smooth. I submitted in 2021, the day before my PhD thesis defense. So it was kind of my thesis project. So I made sure I definitely get it out before I defended my thesis. And the paper got sent out for review like on the same day I submitted it. So after I defended my PhD, I was like, okay, let me just check the status. And I checked, it was already sent out for review. So the Nature editors were very, very efficient.
Yeah, we all see stories of them asking you to do so many experiments over years and years, and it's kind of sometimes not fair. That's why I ask.
The reviews also came back positive, and it was not. They didn't give me a hard time. It was really just. In the final step, when the paper is scientifically accepted, they still give you a long list of things like formatting, making sure the figures align with Nature style, making sure the formats are all correct. That took me some time. And if you notice this paper, all the colors of all cell types are coordinated throughout the paper. Yeah. And I don't know if you checked out the supplementary figures. So we have main figure, extended figure, and the supplementary figures.
I was wondering about that. Can you comment on that? Like, what does that mean? I saw that there's the main figure, the extended figure, supplementary figure. So it's not something I commonly see in papers?
Yeah, I think for most papers, it's main figure and supplementary. In Nature, they do have extended data figures, which is part of the main PDF. Right. So initially I wanted to, so I submitted a paper with 20-something extended figure and 5 main figures. So it was like 30 figures. Each one had like 10 panels. So it was like 300 panels of figures. I had to coordinate the colors, and also like cytokines are in Greek letters, right? That's the proper name. I had to figure out how to make sure every single figure has like Greek letters and listed in the same order, and the font size is like aligned with the requirement. That took a long time, so I initially submitted 20-something extended figures, but then the editors were like, no, we have a limit of 10 extended figures. This is a huge paper, so we can give you 2 extra extended figures, but that's it. The rest of them, you have to move to supplementary. Yeah, so that's why you see a lot of supplementary figures as well. Please definitely check out those figures. They are very information dense. We treated those figures just like main figures, and those are very. I hope they contain important information there.
The figures are unbelievable. Some of them are hugely complicated, and I can't imagine how long it took. For example, I'm looking at figure 1D. Oh my gosh. I mean, just, you have to come up with a design first that presents the data, and then you have to execute it. Did you do these yourself, or others did it?
Oh, yeah. I made every figure myself in the paper. Yeah. So figure 1D is indeed a very challenging figure to make. You asked me why it took like 2 years in total. There were a few months just from figure 1D because it's very. So the analysis at the end, the final results, the analysis are actually not terribly difficult. Like we basically just computed number of genes, and then we computed a degree of the magnitude of response. But it took a lot of time to design this figure to make sure it's all representing the information correctly, accurately, and in a very intuitive manner.
Can we step back just a little bit? So the basis of this paper is you said there's 86 cytokines, and you injected each cytokine individually into 3 independent mice, right? Then a couple of hours later, you collect the lymph node and do single cell sequencing on all the individual cells. So basically, the dictionary is that it is the individual response of individual cells to one individual cytokine, and then look at 86 different cytokines. So this is what we're talking about. This is a huge amount of data. And so representing, you had 380 some thousand individual cells from all of those experiments. And then you got to try and figure out what information is in there, right? So how do you decide what the easy questions are, what the important questions are, and how you do this? So for figure 1D, you're kind of trying to describe the response of the individual cells to these individual cytokines as far as how much of their genome changes and their transcriptome changes. So differential expressed genes for each cytokine for each cell type. So how do you figure out what's the low hanging fruit and what are the important questions and which ones you just have to catalog? And
that's a great question. It's true. The paper has the data has so much information, and to just figure out what data to present, that itself took us some time. And so for figure 1D, even though I felt this figure was challenging to do it well, I really wanted to have that figure there because it is very important. So I think this is, as I said earlier, just having the in-depth experience and knowledge in the field of immunology, even though I originally received training in the computer science, it's very, very important to have this deep insight. So I knew that having this map, just having the understanding of which cell type could respond to which cytokine. We just never had this map before, but this map is so important. In textbooks, for instance, we always talk about just a couple of diagrams, a couple of cytokines, and a couple of cell types responding to cytokine. So it was clear that if we could have a map of how every cell type responds to every cytokine, that would be very useful. And in the first figure 1D, we didn't go deep into the specific genes or anything like that, but just even knowing which cell type can respond is already very valuable. So that's why we decided to put figure 1D there. And so another question, you're why it was hard to make and how do we resolve it? At the end, one thing that was really hard for us was different cell types are not necessarily comparable. A strong response in T cell versus a slightly weaker response in B cells, that does not necessarily mean the T cell response is really just stronger than B cell. It could just mean different cell types have different properties. Some cells just express a lower number of genes in general. And so different cell types is just really hard to compare with each other in a very direct way. So at the end, we decided to give a color ramp for every cell type. So just to guide the readers a bit more. So just because one square is bigger than or darker than the other one, it doesn't necessarily mean it's necessarily a stronger response. It could be, but it's not necessary. So, just simple changes with color could already help a lot, but it took us a long time to come up with this idea. So to decide what information to present in the paper, figure 3 is a figure I like very much. So figure 3, we talked about cell polarization states. And this is based on the initial discovery of macrophages could be polarized into M1-like versus M2-like states. But we immunologists all know that macrophage polarization is much more complex than simply M1, M2. And new states are being discovered, and it's also not. So So I thought, because we have this data now, we can first define the macrophage polarization a bit better. Mm-hmm. But the bigger question is really, what about other cell types? Do they polarize in response to different cytokines at all? Or do they simply turn on or turn off? And that's other cell types can do. Because before building this dictionary, my impression, just by reading the literature, is that only macrophages polarize. Other cell types can do a little bit different things, but it was not crystal clear. But by building this map for every cell type, we learned that every cell type responds to cytokines in very diverse ways. And the macrophage polarization is not more or less than the polarization of other cell types. Mm-hmm. And we systematically listed all the polarization there. A specific example we showed was for NK cells. Like for NK cells, we showed 4 polarization states. But if you look at the extended figures, you can also look at the cytokine responses to every cytokine out there. So for the 4 major polarization states, we found that the interferon state, which is kind of expected, so it shows a lot of ISG in response to interferons. And we know NK cells become cytotoxic when treated with IL-2, IL-15, or IL-12. So these 2 states were kind of known in the literature. But we found 2 other states. One is NK cells in response to IL-1 alpha or beta. And this state does not overexpress the cytotoxic molecules at all. And instead, it overexpresses genes that encode a receptor for interferon gamma. And this just shows that NK cells do not when they activate, they do not necessarily only express the cytotoxic molecules. They're probably, we don't know what the state does yet, but it's probably sensitizing NK cells in a certain way. And another state was triggered by IL-18. Based on literature, I really thought that IL-18 activated NK cells would be more or less similar than compared to IL-2 or IL-15 or IL-12 activated NK cells. But once we got the data, it was very shocking to us that IL-18 upregulated like 1,000 genes, whereas other cytokines only upregulate like a couple hundred. And a lot of the genes in the signature are different, very different from other cytokine-activated NK cells. So using big data approach, we really learned a lot of new things about cytokine responses and plasticity of immune cells that we didn't know before.
So how did you decide to look in the lymph node and sort of where to deliver the cytokines? I could imagine lots of ways you could have chosen to do it. So why did you choose the way that you did, or the time point 4 hours?
Yeah. So we had another project that I started earlier, actually, to look at the longitudinal response to vaccines. So my advisor and my other mentor, so Dr. Nir Hakohen and Dr. Catherine Wu, they developed the personalized neoantigen vaccine. Back then, it was they run one of the first clinical trials to demonstrate efficacy in the melanoma patients. So back then, we were very excited about that result. So I was studying more on the mechanistic side of vaccine responses in mice. So for vaccine responses, lymph node was the natural organ to go for. So we inject it under the skin and we analyzed the lymph node. And so it's from that study that I learned that lymph nodes are more diverse. And it seems that using the system, we were able to deliver whatever we're injecting into the mice to the lymph node robustly. But we still need to test cytokines. In theory, it should get there as well. It's the draining lymph node, but we still needed to test it. So we used interferon. We knew interferon induced ISG in every cell type. So we injected interferon. We did some pilot study. We looked at lymph node cells, and we saw every cell type responded to interferon. So that's how we knew that the system can work. And we also provided one of our extended data figures. You can see we showed that interferon signature can predict PBS versus interferon treatment for every cell type. So this is also a common question people ask, like, lymph node structure is very complex. How do you make sure the cytokine actually gets to everywhere? But we did make sure that the cytokines got everywhere.
And interferon is an interesting one because we expect most cells to be able to respond to interferon. But were there surprises in your data to you about which cells responded to what, or how many different cells responded, or anything like that?
There are a lot of surprises. So the NK cell was a major surprise to us. I can think of at least a few more. So IL-1 beta was very interesting. So IL-1 beta is like a pro-inflammatory cytokine. We expected the responses to be somewhat similar between cell types, but we really saw a highly cell type specific response even to a single cytokine. And though we do not exclude the possibility that IL-1 beta triggered the release of other cytokines that induce the cell type specific effect, but we couldn't find evidence, at least from our data, that any other cytokine induced the same response. So it's very interesting. We're still following up on that to see why exactly that happened, but it shows that immune cells, even in response to a single cytokine, can perform different roles. And another one that I was very surprised is the response to IL-2. So IL-2 is a very important cytokine. It's also a therapy. So it's been used as the cancer immunotherapy since the 1980s. But even to date, there has been so many challenges of bringing IL-2 to the clinic. And that is because the therapy has a very narrow therapeutic window. There has been a lot of effort to engineer IL-2 to bias a specific cell type, like bias towards CD8 T cells and not away from Tregs and all of that. But when we did this experiment, we were surprised that IL-2 was not only activating T cells, it was activating every cell type out there. And we were like, is this real? Can this, but we checked all the replicates and this is clearly what the data is showing. So we looked deeper into it and we learned that IL-2 activates NK cells, which is known. And then every other cell type expressed the interferon gamma signature. And the interferon gamma signature is something we can simply look at the interferon gamma part of the dictionary, right? So this is one of our extended figure for C to E. We showed that IL-2, IL-15, IL-12, and IL-18 can induce the upregulation of interferon gamma gene, and every cell type overexpress a signature for interferon gamma. So this just shows, this is another surprise to us because it shows that the in vivo effect to a single cytokine is much more complex than what people typically consider. Like we so far the field is mostly considering the primary effects, even though we know it's in the body is more complex. And we demonstrate that the body effect is indeed much more complex than the primary effects.
So technically, I'm curious how you did this. Did you do one cytokine at a time? Did you multiple or all at once? Tell us how it worked.
Yeah.
I think the data had we did an experiment across 14 different runs. So for each run, that was between 20 to 30 mice. And it was a lot of work. These experiments are quite painful because it's a very long day when you're running those experiments. So technically, start a day at 11 p.m. to prepare cytokines. We wanted to make sure all cytokines are used fresh and carrier-free. So we reconstitute cytokines either the day before or right before we started doing the experiments. And we need to make sure we give it a sufficient amount of time to dissolve and everything. And we then do the injection at 2 a.m. As I wrote in the paper, the data collection happens between 6 and 8 a.m. for every single experiment because the circadian clock could influence transcriptomic signature. And because I knew I would be the one ended up analyzing the data, I wanted to make sure the data is absolutely the best quality possible. Yeah. So even though that was a long day, I was like, this still needs to be the best quality. And we did it in a very consistent way. So I was already working so much with mice. I got very proficient with analyzing, extracting lymph nodes. And I guess like the actual doing experiment, we did it as quickly as possible, but it's because we are already doing a lot of work with that, and I was already like very proficient with the experimental procedures.
How many cytokines would you do at once, though? Like when you inject them? So
it was 30, around between 20 to 30 mice, so 20 to 30 cytokines at once. Yeah.
And so very quickly you get all the injections. So would you do consecutive days, 20 or 30 cytokines then?
Some of them were consecutive. And others, I was like, I just need a break. It's a long day when doing these experiments.
You're not going to do overnights multiple days in a row, no.
But in theory, you could have all the data relatively soon, and then you just have to analyze it, right? Did you ever have to repeat, for whatever reasons, injections?
We never had to repeat, but we did do replicates.
Right.
We made sure that we had at least 3 mice per cytokine. Right. Just even for single-cell RNA-seq, it's very important that we do replicates. It's because you cannot trust single mouse results, right? Sure. So after we got the first batch of data, we look at the data, we confirm the quality was very good. We decided that we are going to do replicates now. So we did the second and third set of replicates.
And so you collected all the data, you do all the cytokines, and you collect the lymph node cells, you get all of the RNA-seq data, and then you subject it to your analysis, right?
Yes.
So then there's a phase where you don't do wet experiments anymore, correct?
That phase, actually, I submitted the experiment for sequencing a week before COVID shutdown started. And I was so lucky. They were able to process my samples right before. And once we got the data, I went back home to Toronto and I analyzed the data.
So all the data for all the cytokines and all the cell types you had before COVID?
We submitted all the samples before COVID.
Wow, that's lucky.
Yeah.
It's quite amazing. So how long did you take to analyze the data?
Let me see. I think I got the initial set of samples January and
had to be like a little over a year before you put the paper in if you submitted in fall of 2021.
Because COVID 2020 and then submit in 21,
a little over a year.
Did you have to write any new algorithms or anything for the analysis, custom, for example, that weren't already available?
Right. Almost all the figures I had to do some custom analyses because so we do use software packages a lot, but a lot of it, designing the best visualization for 86 different cytokines, that itself took some time. That's true. And also we developed the companion software, immune response enrichment analysis, or IREA. That is for other people, other scientists to use our software to better understand their data. So how it works is basically compare user data with the immune dictionary data. And because we have 86 different cytokines for each cell type, we can simply compare user data and see which cytokines are most likely active in the user data. So, in the past, for example, if people wanted to understand which cytokines are involved in like immunotherapy response, for instance. They would, knock out one cytokine at a time, and they could make some hypothesis based on receptor expression, but it's not clear, right? So now we have this tool. So anybody can basically use our tool to compare their data with the dictionary data, and it can make predictions automatically. And we already run our software on multiple diseases. We did it for cancer. We did it for lupus. We did it for HCV. So my HCV paper actually was also published recently in the Journal of Hepatology, and we're also very excited about that story. And so that one, we used some of the immune dictionary data, and we used the IRIA software to run some of the analyses. And finally, we also did it for COVID. Yeah, and the software just automatically, the top cytokines predicted by the software are automatically the most well-known cytokines associated with each disease. So that makes us very happy.
So basically, you could look at gene expression patterns and ask which cytokines are doing that, right?
Exactly.
That's pretty cool. And you could,
and it's not just for one cell. You're using the fact that you're looking at a whole bunch of different cells and how they respond, and that helps you figure out what cytokine it is, right?
Exactly. So
it'd be hard with just one cell.
Exactly. Because we had a whole bunch of cells, we could do statistical analyses.
So in fact, a lot of people have collected this kind of data for various conditions, and now they could go back and look at it and say what cytokines are involved, right? Even with, for me, infectious diseases would be really interesting, right?
Yeah, exactly. And, so this was inspired by pathway enrichment analysis, Go or gene set enrichment analysis. But I don't know, like immunologists have been using it. We find it a little bit frustrating. It's helpful. It does help you understand the biological processes, but I don't know if you've run it before. Basically, I start with an immune disease or immune process. And then I do a lot of data collection. I run it through pathway enrichment analysis and tells you your data is enriched for an immune process. Your data is an inflammatory disease. And I knew it. But now we can really use this immune dictionary to get much higher resolution of which cytokine pathways are triggering the response you're observing in the data.
So what kind of data does a user need to put into the system?
The simplest one is just a set of genes. So we support that. It's basically the same input as pathway enrichment analysis. You can do a differential gene expression analysis. You obtain a list of genes. On the web portal, we even supply the example. So if you just click on example, you can see that you enter a list of genes, you press submit, and in 30 seconds, you get the results. So our software does not only show which cytokines are most active, it also shows the immune cell polarization states on a radar plot. As I showed in my paper, figure 5C, we showed how macrophages polarize in response to, PD-1 therapy, and the software automatically predicted that macrophages polarize into M1-like, which we call the MAC-B state, which is induced by interferon gamma. We know interferon gamma induces M1-like macrophages, and it polarizes away from the IL-4-induced MAC-E state of macrophages. And this radar plot really helps picture the polarization states because it's not black or white. It's not like M1 or M2. You see a quantification of how much MAC-A, how much MAC-B, how much MAC-C, and DE. And you really get a sense of which state the cell is more biased toward.
Is it possible for someone to kind of look at the data in the dictionary to generate hypotheses?
Oh, totally. Yeah. So now, for instance, the immune dictionary showed a lot of responses that were not characterized before. So we can simply look up the dictionary. Like, for example, if we see a response that has never been characterized before, we can hypothesize. And we knew that the cell type or the cytokine is involved in some disease, then we can hypothesize that maybe it's this cell type, this cytokine interaction that led to a specific disease or immune process. So I think it generates a lot of new hypotheses. Or we can look at a specific cytokine signature for each of these 1,400 cytokine cell type combinations to generate new hypotheses on new functions of cytokines. As I talked earlier, like how NK cells may have new functions besides just the cytotoxicity. It can also upregulate a range of other genes. So these are all the hypotheses we can generate from the dictionary.
Do you think that the genes induced by any particular cytokine in a specific cell are the same across different species of mammals, for example?
Mm-hmm. Yeah, that's a great question. So, we get this question a lot, like a difference between mice and human, and also things like how about different time points? Like, if we measure at different time points, it's possible that not all the responses we're seeing at 4 hours is the same. So I think there are definitely differences, but also the mouse immune system is actually very there is a lot of similarity between mouse immune system and human immune system. So because of this, a lot of reference databases for the immune system were built from mice. Like, for example, the Imogen project is entirely mice. And so even though there are differences, a lot of therapies were also discovered in mice, like the PD-1, anti-PD-1 therapy. It was discovered in mice, and it turned out also works in humans. So while we think there would be some differences, we think there are also a lot of similarities we can learn.
Because obviously if you're using your algorithm to say what's happening in a human when they're given, if they're infected or so forth, obviously you have to assume that there's some similarities, right?
Yeah, so we look at a homolog database to map the mouse and human genes for that purpose. And when we run the software, actually some of the diseases that I just talked about, we collected from human data. And the software and the immune dictionary also automatically predicted the most important cytokines there.
I guess you could also look at immunotherapy, right? If you're using, say, tocilizumab, right? Which is going to, IL-6 knockout, right?
Yep.
Then you could see if the genes are off that you would predict would be off, right?
Exactly. Yes, we can look at the responses. And that's IL-6 is really interesting. We did not see much signature to IL-6 in our data. And that was quite shocking to us, but we did do the experiment in 3 different mice in at least 2 different orders of cytokines from reputable vendors, like Peppertech and R&D. Those are very quality places. We didn't see much response. So we talked to IL-6 experts, and they said it's you need something to get the system going to have an IL-6 response. So in the native lymph node is probably just not the right environment. But to answer your question about seeing the responses to therapy, that's right. Like if there is, if the therapy is known to block a specific pathway, then you can check whether the signature is, downregulated after the therapy.
I'm looking at IL-6 on figure 1D, and there's no response.
It was so shocking to us. I don't know. Yeah. So I think
because that would be a nice validator. What else is given to people? What other cytokines are given to people that you could use to validate?
Oh, IL-23, isn't that the IL-17? Those are newer ones.
So 23 had a response in lymphoid cells. Yeah.
What else?
Well, certainly interferon beta has been given.
Oh yeah.
Anyway, it would be interesting to get samples from those trials or patients just to validate that everything is the same or what, because that's the best you can do, right?
Yeah.
And you'd have to use blood cells, right? Because you can't take lymph nodes out of people.
Can, but they're not so willing. People will do it.
Well, if you haven't removed that surgery, then you could do that. But yeah,
they can do biopsy, but that's very few cells, right? Yeah. You need more cells than that.
So you mentioned a little bit about the idea that an IL-6 response maybe needs something else to kind of get the system started. And so I was wondering how you use the database or the dictionary to think about what might happen in the case of multiple cytokines, because that's one thing. You're delivering individual cytokines here. So, what are your thoughts about what would happen with multiple cytokines interacting with one another, and what does this tell us about that?
Oh, totally. And we know cytokines may have synergistic effects with each other. And typically in a body, cytokines don't come one at a time. They come together. And that's why we have the cytokine storm in a lot of diseases. Like the most well-known one is COVID, right? The severe COVID-19 patients have cytokine storm, which is why the symptom is so severe. So this is so how we thought about it is initially the first version of the dictionary. We want to be able to really just separate out the effect of each cytokine. And that's why we decided to do just one cytokine at a time instead of doing combinations. And even looking at the cytokines in the native context without any other type of environment to get it going, we were already seeing a lot of responses to cytokines and very diverse responses. So this is the first step that we're thinking to start building a comprehensive map of be able to predict every combination out there. And it's also a reason why I started my lab, so I can start keep building this framework. When we go to combinatorial problem, the first dictionary was already stretching our limit quite a bit. Yeah. And we are thinking about, expanding this framework, but also need to think about what's actually feasible to do.
Yeah. Well, I think that also, I could imagine that I could look in a textbook and say, oh, well, these cytokines tend to be made together. So that's a combination we've been thinking about. But I think that your data of what are the products that are made downstream of one cytokine may actually give better information about what the combinations should be. And so I think that I would want to see this kind of data first to learn what combinations really come together, as opposed to just sort of textbook definitions.
That's right. And actually, that's exactly right. So when I run the first 86 cytokine study, I actually included two combinations. The first one is IL-2 plus interferon gamma as an example of textbook knowledge of activating two very different pathways. So how can we learn from that? Another example is IL-2 plus IL-15, textbook example of two similar pathways, and to see what we can learn from the data. And exactly what you said, what we learned at the end is that IL-2 induced interferon gamma. So the IL-2 plus interferon gamma response looks pretty much just the same as IL-2 response itself. So that's exactly the reason why the first version of dictionary is also already so valuable, because you already learned the cascade effects that you don't typically think about.
Do you think that it's possible that some combination of two or more cytokines would give a completely different gene expression profile than either cytokine alone? Is that possible?
So I don't my hypothesis is that when we have combinations, it's going to be response from each cytokine alone, and there is the part that is synergistic between the two cytokines that you cannot capture by individual cytokines. That is my prediction, but I don't have the data to back that up yet. Is it possible that the response look completely different from individual cytokines? I mean, I don't exclude this possibility, but we also checked that some of the just well-known cytokine responsive genes like, IL-4, IL-1 respond to IL-4, TNF, AIP3 respond to TNF. So everything that people have, discussed before, we were able seeing the signature in the data. So yeah, I would guess it's good. There's going to be combinatorial effect, but it's not going to be completely different from.
Understood.
Yeah.
I think an interesting set of combinations would be with inhibitory cytokines like IL-10 and TGF-beta. Do they actually suppress the same for all of the cytokines, or is it different for different cytokines? And even for the same cytokine in different cells, do they respond differently? I think that would be a really fascinating question.
I agree. Yeah, I totally agree. That's a lot of combinations, but that would be super fascinating. I actually thought about initially doing IL-10 instead of IL-10 by itself, but do IL-10 with like a pro-inflammatory cytokine to see that. In the end, we were just like, let's do IL-10 alone, see what happens. And it turns out IL-10 alone also has an effect. Yeah. It induce a very different state of dendritic cells. We show the polarization state of IL-10 induced dendritic cells in the data, and later, there's been literature documenting how IL-10 pre-treatment of dendritic cells can lead to a certain response. So it made sense that IL-10 alone also had an effect.
So are there limitations with that though? Like, do you think there are potentially states that you could see in your data that might not actually exist in reality because those cytokines would never be encountered by that particular cell in isolation?
Yeah, so because cytokines never encounter cell types never encounter a particular cytokine. Yeah, so in the tissue, the cell types that can receive cytokines from surrounding environment, and if the surrounding cells do not produce a particular cytokine, it's possible the cell type just will not receive it. But at the same time, the immune cells travel throughout the body. And, at least, so we also made a cytokine production map, and even among the immune cells, we found that almost like among the 86 cytokines, I think more than 60 of them, we can trace the expression of those cytokine genes encoding those cytokines in the immune cells or stromal cells, like fibroblast reticular cells and endothelial cells. So, we think cytokines are just so ubiquitous and they diffuse very quickly. Yeah. So it's very likely that many immune cell types would be able to get exposed to many cytokines.
Yeah, I think that piece that you just raised about the FRC specifically, I thought that was fascinating, that you could then trace back like which immune cell makes which cytokine. Mm-hmm. Because people have looked, at RNA-seq data and whatever, but really you're mapping it not only to this cell produces this cytokine, but then you can map that cytokine based on your data back onto cells. So you can connect the one cell type with all the other cell types it could communicate with and through which cytokines. I thought that was great because I always tell students when I'm starting to teach immunology, I'm like, it's not linear, it's a web, and it's really complicated, and we'll teach you some connections in this web, but there's no way we can teach you all to you. But then when I saw your data, I was like, it is way more complicated than I had ever even, the web in my head was a certain set of connections, but yours is even orders of magnitude higher than that. And it really, I think, hammers home the interconnectivity of immune cells and how they communicate with each other to orchestrate responses. It's just fascinating.
Oh, completely. I completely agree with you. And I think I know the diagram you're picturing, because we also see those cytokine network diagram. If you just Google the cytokine network diagram, and I show this diagram to my friends now studying immunology, and they're like, "This is so complicated." And I was like, "Oh no, this is not even 1% of the reality." Yeah, and we built the cell-cell interactome, as we pointed out. That's when we realized the interconnectivity in the immune system is huge, and the immune system really have to constantly talk to each other to orchestrate any response. One of the lessons, just by building this dictionary, I just felt so fortunate that our immune system is working very hard for us. Like, how does it even work? Like, there are so many cell types, so many cytokines, and they all need to work correctly with each other. Like, how is that possible? Like, we're very amazed.
It's evolution, man.
Yeah. and all these different stimuli will change expression of the receptors that then can change the response of that cell to that cytokine, which is just
Exactly.
What I think is amazing is this IL-1 beta, which does something different in every cell.
I know. I think that was the most surprising thing to me.
That's amazing.
That was very surprising.
Figure out how that might work, right?
It's the same receptor.
Yeah, same receptor.
Right? So it tells you if you study signaling pathways, throw your hands up in the air. Like, I don't know what to do with that. We study a signaling pathway in one cell, but then it's totally not, translatable to any other cell.
It can be different in a different cell type. Yeah.
Yeah, I know. It makes me think, like, are there any unknown receptors that haven't been discovered? Because like, if it's one receptor, why is it so diverse?
Yep.
Like, yeah, or maybe the cytokine induce like other types of like molecules that's not a cytokine, because we study cytokines pretty exhaustively. We did not see anything else that looked at the IL-1 beta response.
Well, that's true. You have a whole nother set of metabolites that are not cytokines, right? That could also influence. And, the cytokines take a certain amount of time to make. And so 4 hours is a logical time frame to me to inject something, to have it get to the lymph node and look for a gene response. And we would consider that in general a primary response, but you're telling us that you're already, for example, seeing an interferon gamma response on top of whatever cytokine you give, like in the NK cell. So even 4 hours is not early enough to capture the primary isolated response. And then,
I think we thought a lot about is it possible to capture only primary effects. We thought a lot about initially we thought about setting up a microfluidic system to flush out cytokines as you make them. We talked to a lot of people and brainstormed all types of wild ideas, but then we realized they wouldn't be physiological as you flush out cytokines. That flushing also creates a gene expression change, right?
Yep.
So
the physical part. Yeah.
So we thought it would be best to just do it in the physiological setting. Even if there's a secondary effect, that is also part of the response. Because if every time you give IL-2, that induce the same secondary effect, it's very important to consider that.
When thinking about some of those effects that you see, did you learn anything about maybe an amount of change in gene expression that actually leads to a measurable change in other phenotypes downstream? Or, is there a certain sort of number of cells that is, too few or too rare to really make a big difference? Or maybe you found, even if you have a very small number of cells producing cytokine, that can be a big difference. Maybe we had overlooked that small number before.
Yeah. As we built a cell-cell interactome, we saw basophil was one of the major players in the cell-cell interactome, mediating cell-cell communication in lymph nodes. I honestly didn't even know that there were basophils in lymph nodes. I don't think anybody reported it. But maybe there are a couple of papers that reported it. And but when we look deeper, we found that basophils do not only express a large number of cytokines, they also express some important cytokines, like IL-4, IL-6. Like IL-4 is important for, the type 2 immunity, and we didn't know exactly how that started, right? So we think that even though basophils are extremely rare, they may be the initiator for type 2 immunity developing lymph nodes.
Well, that's amazing. Anything else, guys, before we leave?
I was just going to ask, what's next? So we talked about some of the ideas of mixing and the impracticality of the magnitude of that kind of the type of experiment. But what are you thinking about? Like what you're going to work on?
I think we're still going to do some of it. It's not possible to do the entire combination, all combinations ever out there, but we'll do some of it. So I started my lab, and we have 3 areas. The first one is to continue developing the immune dictionary to make it a powerful platform, both from the data side and from the software side, so we can predict like any immune response. And the second one is to follow up on some of the hypotheses generated from the dictionary, for example, to characterize new functions of cells or characterize how specific cell subsets or states can play a role in disease. And the third one is to collaborate with clinicians to understand, use such methods, big data methods, to understand the different diseases. We're very busy. There are so many projects, but even just from the dictionary, we immediately can think of several follow-up studies there.
So I feel like I have to ask this question on Steph's behalf, as she's not here with us today. But have you done any comparisons between delivery in the skin and delivery at other sites, particularly, as she would ask, mucosal sites?
We so far only deliver through the skin. But yes, I think it could. I mean, delivery is known to modulate like vaccine response, right? So I wouldn't be surprised if there's a different immune response to the delivery site. I think like the mucosal delivery would be very interesting. It's
I was especially wondering with things like oral tolerance and all of that.
Right. Yeah. So we're thinking about different delivery sites as well. Like we should, we'll think about doing a comparison. Yeah.
So do you have any computational biologists in your lab who you are teaching immunology now?
Oh, so many. Yes. Yes. So our computational biologists all have to learn immunology, and immunologists all have to at least know some computational concepts so we can communicate with each other.
What do you think are big computational concepts that an immunologist should know?
Yeah.
So as an immunologist, what should I learn?
Yeah, exactly.
I think immunologists already know so many computational concepts. Like, for example, the TISNI map, TISNI is actually a machine learning algorithm. So as I mentioned, I'm from Toronto, and Toronto is a major hub for machine learning. So the TISNI map was originally developed in Toronto by our senior faculty, Dr. Jeffrey Hinton, who also pioneered the deep neural network, which is the computer architecture that's in almost every AI ML algorithm these days. So this is a technical machine learning algorithm. People don't think about it often. And every immunologist know how to look at the TISNI map, how to look at different cell clusters. It's actually surprisingly intuitive once you generate a map like this. So I think we just need to... So I think immunologists already know a lot, to be honest.
Well, we could definitely think non-linearly, and that's helpful in analyzing a lot of the data.
Yeah. So yeah, definitely. So one thing, I guess, it's the data has more than 20,000 dimensions representing 20,000 genes. And the one we projected onto 2 dimensions is never going to be perfect. Like just by definition, it cannot capture all the high dimensional data. So there will be some distortion. You will definitely see that. So I think maybe that's one thing to keep in mind. Like when we look at 2D maps, always know that it's not reflecting every information out there in high dimensional space. But I think immunologists already know that we use 2D maps mostly to gain intuition. It's still super, super valuable to gain intuition that's not possible to do without having a way of visualizing the data.
All right, that's Immune episode 75. You can send your questions and comments to immune@microbe.tv. And as I said before, if you like what we do here on our science programs, consider supporting us. Microbe.tv/contribute. Our guest today from Harvard School of Dental Medicine, Ang Sui. Thanks so much for joining us.
Thank you so much. It's my pleasure.
Great story. Immune dictionary. You do have a dictionary in your office that you can hold up, right?
Definitely.
Cindy Leifer is at Cornell University. Cindy Leifer on whatever. Thanks, Cindy.
Thank you. This was great.
Brianne Barker's at Drew University. BioProf Barker on. Thanks, Brianne.
Thanks. This is great. I learned a lot. Apparently, I must know something about computational biology that I didn't know before.
Yes. The things we know that we didn't know. I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.